So hello, everyone. So in this project that I will talk about today, we were looking at how we can modify life history trait changes, or how we can modify life history traits in phages, and how this actually impacts the evolutionary success of a phage. Here we go. So for our study, we used the model phage Phi-X174, which is a strictly lytic phage that doesn't have a tail. It has 11 genes, and it infects E. coli C. When we measure in our lab, it has an approximate lysis time of 17 minutes and a burst size of approximately 140. So because we come from an evolutionary biology institute, of course, we used experimental evolution to actually change those life history traits. And the way we did this actually is using a serial transfer approach. So in our experimental setup, or in our basic experimental design, we always propagated the phages onto susceptible hosts to avoid any co-evolution between the phage and the host and just have the phage evolve. We use an MOI input of approximately 0.1, and we do those serial passages in a liquid well-mixed environment. So the idea was basically to select for specific life history traits and then afterwards check which traits are under selection and how they change in which direction. So we established two different transfer regimes that basically just differed in the time that the phage is allowed to infect the bacteria. In the 30-minute regime, we propagate the phages after 30 minutes, which means that we don't limit host availability, and the phage more or less is able to complete one round of infection. Whereas in the 3-hour transfer regime, the phages run out of host pretty quickly throughout the transfer time. When Phi-X174 lysate is plated in a plaque assay, it forms large clear lysis plaques. And our evolved mutants in both transfer regimes, after a couple of transfers, actually display opposing plaque phenotypes that are actually heritable phenotypes. So in the case of the 30-minute experiments, the plaque size increases by approximately 1.5 times, whereas in the 3-hour transfer experiment, we see a drastic reduction in plaque size. In both cases, approximately 20 generations are required to actually isolate those mutants. So now we looked actually what are the genetic basis of those differences in plaque morphology. And what we found is that for both transfer regime, one single point mutation that differs from the ancestor in the major capsid protein is responsible for the respective phenotype. However, the position for the two transfer regimes, one you see here in purple, one you see here in orange, is different. And when we look at the mutations actually in the major capsid structure, what we found is that the mutations that lead to the small plaque forming phenotype always occur at interfaces between monomers. Whereas the one that leads to the large plaque mutant is more towards the center of a protein monomer. So our first idea was, okay, if these mutations are located at monomer interfaces, maybe the capsid stability and therefore the extracellular persistence is impacted. So what we first did is we measured the fraction of infectious phage particles over time for the specific mutants and for the ancestor. The solid lines in gray shows the ancestor, and purple shows the large plaque forming mutant, and the dashed lines in orange and in green show the small plaque forming mutants. And what you can see is that we see more or less a 10-fold increase in persistence in the small plaque forming mutants, meaning that the trait under selection in the long transfer regime was actually persistent. However, the single point mutation, this one single point mutation in the major capsid, also impacted another life history trait, and this was the adsorption. So when we measured the adsorption constant, again, of the mutants and the ancestor, what we could see is that this mutation also increased the adsorption constant of the respective mutants. However, we were quite surprised to find actually that the mutant that was isolated from the short transfer regime showed a 5 times reduction in adsorption constant. And so we were a bit intrigued by finding this, and we were thinking, okay, how can we explain this? Because we did both transfer regimes in a liquid well-mixed environment where fast adsorption should always be beneficial because it allows technically an earlier initiation of infections. So I brought a schematic figure on how to explain under which conditions slow adsorption can actually be beneficial. So let's assume we introduce two different types of phages in our system. One is a fast adsorber, one is a slow adsorber. Both of them in the 30-minute experiment will go on and infect for one round of infection. As I told you, our lysis time of this phage of the ancestor at least is approximately 17 minutes, which means that after approximately 17 minutes, the infection, the first round of infection will be completed. And so now fast adsorbers, which have also not only attach faster, but also have a higher affinity to attach hosts, will go right away and infect the next round of susceptible bacteria, whereas the slow adsorbers will take more time to do this. So at the moment of transfer, which is 30 minutes, the phages, the fast adsorbers, will not be able to complete another full round of infection. So the majority of phages will be associated to the hosts at the moment of propagation, whereas slow adsorbers will initiate infections at the first and the second infection cycle more slowly and will be present at higher rates as a free phage. So when we actually now transfer, those free phages that are propagated can initiate the next round of infection, whereas the fast adsorbers actually are lost either to infections that are present in the second infection cycle of the first transfer, or also because of an increase in MOI throughout the first infection period actually will be present as multiple adsorbers to one host cell. So there is a high rate of phage particles being lost to dead-end infections. As you can see, this is quite tied to a specific experimental condition, which is our 30-minute transfer experiment. And basically what our experimental evolution is showing is that a benefit, or judging if a life history trait is actually beneficial, is highly context dependent. And so in our case, when we have host limitation, which is what we have in our long transfer experiment, high adsorption is very beneficial and is accompanied by an increase in adsorption, whereas the slow adsorption is more or less only beneficial when there is transfer, where there are short transfer times that only allow for one round of infection. Yes, and with this, I would like to say thank you to all the people that contributed to this project. Of course, there's much more to the story than what I just told you. So we also collaborated with our theory department of our institute for mathematical modeling. And if you're interested in any further parts of the story, I would be very grateful for questions and also just coming up to me and talking to me. And there is one specific thing I would also like to acknowledge, which is my group. Because my previous first supervisor left actually last year to transition to industry. And those people, they just adopted me basically and gave me a really, really amazing home. So yeah, thank you. And thank you very much for your attention.